Okay. So hi everyone, hope you are fine. My name is Karina Ladeira and I am the young investigator representative of the International Comet Assay Working Group, which is a specialist group of the European Environmental Mutagenesis and Genomics Society. This is the third Meet the Experts, and the aim of this initiative is to dynamize the dialogue between mainly young researchers, but not only, who use or have interest to use the Comet Assay and experts in the Comet Assay. I have some information that I have to tell you in the beginning. So this meeting will be recorded, streamed, and shared in YouTube, so you can revisit as much as you want in the YouTube channel of the ICAWG group. Also, if you explore our website, you can see that you have there the last 2 Meet Experts that we have already organized. And also there are more technical videos about the technique, so you are free to see that videos and understand the good procedures to make the Comet Assay. Regarding to the dynamics of this meeting, so we will have a first expert presentation that is Professor Andrew Collins, and following his presentation, we will have a space for discussion. This discussion can be like you raise your hand and you directly do the question to the expert. Also, you can type in the Zoom chat, or if no questions rise, I have also a list of the questions that you already dropped when you did your registration in Google Forms. So I will ask also that questions to our experts. And I think is that this about the information that I would like to share. So I will introduce now Professor Andrew Collins, and if he wants, he can start already sharing his presentation. The presentation that I have about Professor Andrew Collins is specifically about the topic that is going to talk today, that is the DNA repair. So I'll talk a little bit about his career in DNA repair. So the interest of DNA repair of Professor Andrew Collins began as a postdoc in Cambridge, looking at the response of cultural cells to UV radiation, and he found that, contrary to the accepted dogma, it was possible to block nucleotide excision repair with DNA synthesis inhibitors. The move to Aberdeen coincided with the introduction to the Comet Assay, and he soon realized that could be used to measure not just DNA strand breaks, but also modified bases, converted to breaks by specific repair enzymes. And that led to the interest in endogenous base oxidation and antioxidation protection. And at the Rovent Research Institute, he applied the enzyme modified Comet Assay for the first time in human nutrition trials. He also developed in vitro Comet Assay-based assay for base excision repair, and these methods were put to use later at the Department of Nutrition, University of Oslo, where he worked until retirement and where he is Professor Emeritus. Most recently, he managed the Cost Action H-Comet, bringing together researchers working with the Comet Assay in human biomonitoring, and actually this working group was raised in that H-Comet action, which I proudly make part of also. And so to me, I'm very happy to have you as one of our experts today. So thank you so much for accepting this invitation. And you can take the floor, the virtual floor.
Oh, I just managed to unmute. So good afternoon, everyone. I'm glad to be here. It's like a family reunion. But I hope there are some newcomers to the Comet Assay and newcomers to the DNA repair assay. So you've probably all read my introduction. I'm going to talk about different ways of measuring DNA repair. but it's worth pointing out that DNA repair was included in the very first publications by Ersling and Johansson and Singh et al. They looked at the recovery of cells after X-ray radiation, ionizing radiation. Anyway, there are a lot of questions still to be addressed, and I hope that during my talk I will deal with them. So one important question is whether the various methods to measure DNA repair are actually looking at the same thing. I want to talk about the variation at a population level, inter-individual variation. And I want to address the question of whether a high DNA repair capacity necessarily implies strong protection against DNA damage, or could it just reflect a high exposure? And then the everlasting question of whether repair capacity increases or decreases with age. So let's go on to the next slide. I don't see it all, but never mind. Okay. So first of all, different repair pathways deal with different kinds of DNA damage. The simplest method is strand break rejoining. Rejoining single strand breaks is fast. It's a question of maybe filling a very small gap and then ligation. Base excision repair is more complicated, but not as complicated as nucleotide excision repair. Base excision repair involves removing the damaged base using a specific glycosylase that leaves a baseless sugar or AP site, pyrimidinic site, and the gap is filled by polymerase beta and ligase. Nucleotide excision repair involves a lot of proteins. It deals with UV-induced pyrimidine dimers and bulky adducts. They're removed in a 28 base long oligonucleotide, and the resulting gap is filled in by different polymerases and ligase. Cross-links are another kind of damage that needs to be repaired, and it seems that nucleotide excision repair is involved in repairing the damage on each of the two strands that are linked together. Mismatch repair. Replication on damaged DNA can lead to incorporation of the wrong base. For example, atoxoguanine, the common oxidation damage, pairs with adenine rather than cytosine. And several enzymes are involved in detecting and correcting mismatches. And then finally, we come to double strand break repair, which is complicated, and I won't deal with it in detail. It involves homologous and non-homologous end rejoining. So just to put DNA repair in context, it is an element in maintaining the dynamic steady state of the genomic stability. And I like to think of it as like a water tank. So there's a constant input, for example, from free radical attack, oxidation damage, and there's this output through DNA repair. So there's a balance between the two, and that affects the level of damage in the cell. If it rises too high, it can lead to apoptosis and cell death. But all the time, there's the possibility of mutation if the damaged DNA is undergoing replication. So individual DNA repair activity is a useful potential biomarker. Not all DNA repair pathways can be measured with the Comet assay. These are the ones that can: single strand break rejoining, base and nucleotide excision repair, and cross-link repair. I'm just going to hurry on. I'm sure all of you know about the standard Comet assay, but you may not be familiar with the enzyme modified assay. So I'm just going to go through it very quickly. After embedding the cells in agarose and lysing them to produce nucleoids, there is this extra step of digestion with lesion-specific endonuclease. So in the case of oxidation damage, we would normally incubate with formamidopyrimidine DNA glycosylase, but other enzymes can be used. And then after that, the rest of the Comet assay is as in the standard assay, and the frequency of lesions is given by the increase in DNA breaks in the presence of the specific endonuclease. For example, the level of oxidized bases is measured with FPG, as I said. So there are three approaches that I want to discuss. The challenge assay or cellular repair involves treating cells with the DNA damaging agent, incubating the cells and monitoring the damage that remains at intervals. So we're looking at removal of the damage over time. And it measures the complete process from incision, recognition of the damage through to ligation. And this can be applied to strand break rejoining and also to base and nucleotide excision repair. The second assay I will discuss is what I call the incision assay. And this can be applied to UV irradiated cells. And it's a way of measuring the normally transient incisions that occur during nucleotide excision repair. And they can be accumulated when repair synthesis is blocked with DNA polymerase inhibitors. The third assay is the in vitro assay, where we prepare an extract of cells and incubate that extract with a prepared DNA substrate of nucleoids containing specific lesions. So we're looking at the ability of the extract, the enzymes in the extract, to recognize and incise at sites of damage. So first of all, the cellular or challenge assay, cells are treated with a DNA damaging agent, incubated, and the damage remaining is measured by taking samples at suitable intervals. So strand break rejoining, you can see here after treating cells with hydrogen peroxide, rejoining is quite rapid. So within an hour, almost all the damage has gone. So that's using the standard Comet assay to measure breaks. Base excision repair in this graph is shown here. After treating cells with the photosensitizer and light to induce atoxoguanine, using FPG to detect the lesions, and removal of this kind of damage takes a lot longer than strand break, so a matter of hours. Second assay, the incision assay, this is not much used, but has potential. And it's really a bit of history. More years ago than I care to remember, I was looking at the effects of UV light in mammalian cells and looking at the level of DNA damage measured, or DNA breaks measured using the technique of alkaline unwinding. So cells were incubated with inhibitors of DNA replication, hydroxyurea and cytosine arabinocyde, before and after UV irradiation. And DNA breaks were measured after 30 minutes. And we found that normal cells, such as these embryonic lung fibroblasts, accumulated breaks, and this is in terms of UV dose. So break accumulation was dependent on the UV dose. Similarly for HeLa cells, but with cells from xeroderma pigmentosum patients who have a genetic disorder in nucleotide excision repair, there was limited or no incision occurring. So at that time, as Karina mentioned, the usual, well, she referred to this, the usual approach to assessing DNA repair was unscheduled DNA synthesis, which involved incorporating tritiated thymidine into the cells treated with the genotoxic agent in the presence of hydroxyurea, which was known to block replication in S-phase cells. And autoradiographs were developed and repair was measured in terms of the silver grains over all the nuclei. So it was generally believed, because repair synthesis was detected in this way, that repair could not be affected by agents that inhibited DNA replication. such as hydroxyurea. But we had observed breaks accumulating after hydroxyurea and UV, which strongly suggested that incision was still occurring. But repair synthesis could not go to completion, so the normally transient breaks remained open. And that's shown here. This is normal repair, rapid process, very short-lived breaks, whereas in the presence of the inhibitors, the breaks turn into gaps and repair is never completed, so the incisions accumulate. So this method could be used to follow the kinetics of repair by pulsing cells for 30 minutes at different intervals after UV irradiation, and that's shown here, 0 to half an hour, 2 to 2.5 hours, 4 to 4.5 hours, and you can see the decrease in incision activity with time, which is consistent with first-order reaction kinetics. Thirdly, the in vitro repair assay, which is the most commonly used now, and it can be applied to looking at base excision repair, for example, in this diagram. We, first of all, prepare substrate nucleoids by treating cells with a specific damaging agent, such as the photosensitizer plus light, to induce atoxoguanine. The cells are embedded in agarose and lysed. We prepare a cell extract from the lymphocytes or other cells that we're measuring, and add that extract to the gel. The enzymes in the extract recognize the damaged substrate nucleoid, the damage in the nucleoids, and create breaks. And the breaks are measured with the standard Comet assay. So we expect to see this result with a control without extract showing no damage, whereas with the extract we see comets with tails. And the rate of accumulation of breaks is a measure of the OGG, in this case, OGG repair activity. In the case of nucleotide excision repair, we prepare a substrate damaged with UV light or benzpyrene, and the rest of the procedure is the same. I want to share some recent results. One of the questions I posed at the beginning was whether the different assays measure the same thing. And until now, this has not been tested, but Jiang, a PhD student in Oslo, has measured base excision repair in a variety of cell lines using the two methods. And just to remind you of the difference between the methods, in the cellular repair assay, we treat cells with damage and look at the ability of the cells to remove the damage with time. Whereas in the in vitro assay, we are preparing extract from the test cells, incubating that with a defined substrate containing damage, and we're looking at an increase in breaks. Anyway, it turns out that applying these two assays to the same cell lines, there was no correlation between the results from the two assays, which might be upsetting, but in fact, in retrospect, it might not be so surprising. The cellular repair assay measures repair after a challenge of DNA damage. So it's looking at the complete process from incision to ligation. And in addition, the challenge of DNA damage might even induce repair activity. Whereas the in vitro assay measures the potential, the enzymic capacity of the cells for repair. And probably the in vitro assay is the more appropriate assay for use in human biomonitoring. But of course, I would say that, wouldn't I? Because I've been involved in a lot of trials where we have used the in vitro assay. But I will stick by that. So this was a study where we had lymphocyte samples from about 30 subjects, sampled on 2 occasions, measured for base excision repair and nucleotide excision repair with the in vitro assays. And you can see these are the 2 sampling dates on the 2 axes. You can see that for each individual, there's more or less the same activity on the 2 days. So there's intra-individual constancy, whereas there's quite a wide range of activity within the population group. And the range is actually even higher for nucleotide excision repair, where some subjects seem to have very little or even zero repair activity. So there's a good correlation between the samples, subjects have characteristic repair rates, and there's a wide range of values for different subjects. So I think that means that this is a good biomarker for looking at intrinsic differences in repair, or the effects of exposure or nutrition, for example. So talking of nutrition, the question, can repair capacity be affected by extrinsic factors such as exposure, occupational, environmental exposure, or in this case, nutrition? This is a trial from quite a long time ago with kiwi fruit supplementation, and for 3 weeks with different doses of kiwi fruit. And we found a strong and a very significant increase in repair activity, basic excision repair activity after the supplementation. We looked at messenger RNA levels for 2 of the enzymes involved, but found no difference. So we don't know how the induction of repair actually occurred. A bit about clinical applications. And the in vitro assay has been adapted to look at repair activity in extracts, tissue extracts, as well as from lymphocytes or other cells. And this refers to an experiment, a trial with colorectal cancer patients, where DNA repair was measured in tumor and normal tissue, and also in lymphocytes. And first of all, there was a slightly higher repair activity in the tumor compared with the normal tissue, but there was a significant correlation between normal and tumor tissue, and also a correlation between lymphocyte repair and both normal and tumor tissue repair. This is a follow-up study where basic excision repair was measured in colon cancer patients. And it was found that basic excision repair in the normal tissue was positively associated with overall survival of these patients and relapse-free survival. So that's a very good evidence of the relevance of repair. Additionally, in a multivariate analysis, good therapy responders with an elevated basic excision repair exhibited better overall survival. Now, we know that mutations in genes encoding DNA repair pathways are among the most frequently mutated genes in cancer. But a separate question is whether intrinsic deficiencies in DNA repair predispose to cancer. And we really need more prospective studies to answer this question, with early measurement of DNA repair followed up over many years to investigate whether there is a link between the early level of DNA repair and the risk of cancer incidence and mortality. I told you, or Karina mentioned, the Comet database, or the Comet Comet action. The Comet cost action. This was set up primarily to collect data from human biomonitoring studies, to create a database which we could then subject to a pooled analysis, similar to a meta-analysis. Most of the results in the 2,000 or so data sets that we got relate to DNA damage, but there were a few hundred studies of a few hundred samples from DNA repair studies. In terms of basic excision repair, it turned out that it was higher in women than in men. There was no overall effect of age, no effect of smoking or occupational exposure. And interestingly, increasing BMI in the subjects was associated with a higher level of base excision repair, shown here, but at a very high BMI, base excision repair actually went down. Furthermore, high base excision repair level correlates with a low level of DNA damage. We've recently been looking at nucleotide excision repair in the same population group, and these are very preliminary results. Again, there was no overall effect of smoking, but in fact, smoking males have a higher nucleotide excision repair capacity compared with non-smokers. And in the non-smoking group, females had a higher nucleotide excision repair than males. But this is ongoing work. So I could now deal with the questions. I'll first try to answer the questions that I asked at the beginning. First of all, are the different experimental approaches measuring the same thing? Apparently not. And just to reiterate, the cellular repair assay is measuring repair overall, whereas, and it covers the complete process from incision to ligation, whereas the in vitro assay is measuring the incision enzyme activity of the cells and their potential repair. And there are pros and cons for the different approaches. One of the problems with the cellular repair assay is that it involves incubating the cells in the lab, so it's not really very convenient for biomonitoring. Sorry, I should go back. How much variation is there in DNA repair at a population level? I showed the graph of the 30 or so sample people who were sampled over 2 dates. Base excision repair, there was a sort of fourfold range of activities, and with nucleotide excision repair, a wider range with some very low levels. And it's not surprising that there is this difference. I see base excision repair as constitutive. It's switched on all the time as there is continuous DNA damage occurring, particularly oxidation. Whereas nucleotide excision repair deals with exogenous damage from UV or chemicals, so it could well be inducible. And if you're not exposed, you maybe have a low level of repair. Does a high DNA repair capacity imply strong DNA protection, or might it indicate that a high level of genotoxic exposure has induced repair pathways? I think there's no clear answer to this. It's probably a bit of both. We do have an intrinsic, genetically determined high repair activity, which probably does lead to protection, but there's probably also induction. And I think the next slide, no, sorry, let's go back. And then does repair capacity increase or decrease with age? There are mixed results, but the overall picture is that there is really no difference, no great difference as you get older. Though this might actually be a survivor effect. So those people who have a high repair potential maybe survive, don't get cancer, so live longer. That might introduce a bias. So I could end there. I could go on to answer the questions that have been presented, but Karina, you would like to ask the audience for questions now?
Yes. First, I would like to thank you for your presentation. And now I would like to ask if someone have any question that want to address directly to Professor Andrew Collins. In the chat, nothing is there. So probably I can start with some questions that were made in the Google Forms, but feel free to ask too, okay? So the first question is which parameters should we use for DNA repair analysis using the Comet analysis program, and how can we make the calculation?
Well, for the Comet assay in general, we recommend percent tail DNA as the best descriptor, because it is, well, I think the best justification of using that is that if you're given a percent tail DNA, you can visualize the comets that are being described. For the cellular repair assay, you could try to measure the initial rate, which would be biochemically the best measure. The trouble is that different cells tend to have a different initial rate after giving them the same amount of damage, and that will affect the initial level of damage will depend on the cell type. So the initial rate depends on the initial level of damage, and so it makes it rather a difficult measure. So we rely on the half-life of the damage, the T-half. For the in vitro repair assay, we need to look at the ratio of breaks over time. All the samples should have the same protein concentration or the same number of cells, whichever is most useful, most easily measured. And adjusting for variable protein concentrations is difficult because there's no proportionality between the repair activity and protein concentration. Meanwhile, we recommend that as far as possible, the extracts are made from the same numbers of cells or similar amounts of tissue. I hope that's an answer. Should we go on to the next?
Yes, the next one is more about, and I'm experienced that we have, because in our Google Forms we have a question of suggestions for future meet experts, and some people are talking a little bit about plants is one of the subjects. So maybe this question I will put in the way, is there any difference in performing DNA repair assay in plant cells in comparison with the animal cells, that it was the ones that you work more?
I think there's not much being done with repair in plants. There are a couple of papers from John Einset, who worked with me in Norway, and he was looking at DNA repair after X-irradiation. But I think if this can be dealt with in the future, that's good. I'd just make the comment that if you're doing any experiments with the Comet assay on plant cells, at least cells from leaves or nuclei from leaves, the cells contain chloroplasts which release oxygen. So the nuclei have to be isolated under a red safe light. Should we go on to the next question?
Yes. This is, I think it is a more general one. In human lymphocyte assays, how many replicates are needed for the Comet assay?
All I can say is have duplicate gels for each sample. I don't know if anyone wants to improve on that answer.
Anyone that have a different perspective?
Can I?
Of course.
The trouble with human biomonitoring is that then you have only two parallels. So what we did when we did it, in that time we didn't use 12 gels, but 2 gel. Actually, this was time where we introduced 2 gel format to a little bit miniaturize and maximize the results, because we needed to have 4 values to do statistics. So I would say that if you have capacity and resources, then 4 would be the best parallel to be able to do statistics.
Okay, when you say 4, is 4 gels?
4 gels, because it's the same human Yeah, or of course you could do fresh blood, fresh lymphocytes and frozen.
Mhm.
Or you could take blood from both arms, have two samples.
No, 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 no. No, but yeah, at least 3 would be really good.
Okay. And the next question, can the Comet assay be used to monitor mismatch repair?
I didn't know the answer to this, so I looked it up and I found one paper from 2005 which uses an enzyme T4 endonuclease 7 to look at mismatches using a cellular type assay, looking at the removal of the, at least I think they were looking at repair of the damage. But I really am quite ignorant about this, so if anyone else has any information, share it now. It's a good question. It's a good possibility.
Okay. We have a question in the chat.
Yes, I will say this. We have a question.
Do you want to?
Andrew, do you want that me read to you, or you can read the question in the chat?
It's better to read, and he read also. So it's from Ali. Thank you for the presentation. Is H2O2 a good challenger when working with whole blood samples? And if so, is there a concentration range to be more adequate? This is one question. I don't know. Then there is another one from the same person.
Hydrogen peroxide with whole blood is not good because, as I recall, there are enzymes such as catalase in the whole blood that break down the hydrogen peroxide. It's okay with isolated lymphocytes, but not with whole blood. What was the other question?
And is also, can I expect to get reproducible results with this challenger if I choose to use it? Or is there a better, more recommended substitute when working with whole blood?
If you're looking at strand break rejoining after hydrogen peroxide, I would say don't do it. X-rays, if you could treat, yeah, you could treat with X-rays and whole blood, I think, and look at rejoining of breaks induced by the X-rays.
Okay, and in a chemical substances instead of radiation. I'm just asking because to have radiation is more difficult.
Yeah.
What about MMS? What about MMS? I think someone tried it.
I remember that we talked already about this.
Yeah.
It was not a bromide, something like that.
Potassium bromate would probably be okay, yeah.
I remember that name.
Yeah, because the catalase would break down hydrogen peroxide, but it wouldn't have an effect on potassium bromate. So yeah, it's worth trying.
Okay, potassium bromate.
Yes.
For whole blood. Lymphocytes, it will work the oxygen peroxide.
Sorry, say that again.
This is me asking. So for whole blood, the problem is the catalase if we use the hydrogen peroxide. But in lymphocytes, isolated lymphocytes, it will work, the H2O2.
Yes, yes.
Okay. The question, it was regarding whole blood. It was just a side question now from me.
Because the lymphocytes have been washed free of the
Yes.
Yeah.
Okay, there is another question, but is a general. So the genes that are involved in different susceptibility in DNA repair is very wide.
Yeah, there are too many to list, I think. But I did look on Wikipedia under human repair genes, and there's a very good article there by Rick Wood and someone else. Rick Wood is one of the leading figures in DNA repair, and there are probably, I don't know, over 100 genes listed or enzymes listed there.
Then another one that I think that is a question that also is more or less addressed it to Maria, but it is here in DNA repair. What would be the best timing for human sampling after an exposure? I think here is directly for the DNA repair, probably, I don't know.
Oh, Andrew,
Well-
would you like to answer?
Well, I'll start. It depends what the question means.
So human
Exposure-
sampling after an exposure.
Well, yeah, exposure of humans is not normally ethical. Though with nutrition, we were, well, with nutrition, you can see an acute effect after a few hours. So giving people vitamin C, you can see an effect after 3 hours. Then there are longer term effects that show up after a few weeks. But if it's a question of taking human cells and treating them in vitro, Maria, you might agree with this, 3 hours for stimulation of enzymes and maybe 24 hours for induction at the level of transcription.
Yeah, and when it comes to human biomonitoring, it's because usually it depends if it is environmental or occupational exposure. So then you sample whenever when you know it's a constant exposure, usually with workers or environmentally. So you sample them in the same. Important is that if you have several groups, you need to sample them in same time, same season, for example.
Yeah.
We did this study, FiberTox study, and we did September sampling, all of them. So just to avoid seasonal variation in background level of DNA damage and repair. But it's because it's kinds of steady state, induction of damage, induction of repair, and then, you know, as Andrew already said. So it could be any time.
Okay. So I move to another question. What is the future application of DNA repair assay?
Well, I would like to see a miniaturization of the assay so that we don't need so many cells for the in vitro assay. Millions of cells are needed. And also, it would be good to be able to carry out the assay on frozen samples from biobanks. And I think the number of cells present in that is a limitation at the moment. And it would be good to see general acceptance of the DNA repair assay as a useful biomarker assay in human studies.
Thank you. There's a question that is more about, is another topic that I would like to address in another meet experts, because of the there are lots of people that start to want to know more about the application of Comet assay and spermatozoa.
Yeah.
So I think we can skip this question.
Okay.
And the other one, how DNA repair assay work during radiation exposure?
I'm not sure what this means, but radiation induces mainly strand breaks.
Mm-hmm.
So the in vitro assay is not appropriate because it's based on the production of strand breaks by an enzyme. But the cellular assay is appropriate for looking at repair of radiation-induced strand breaks.
Okay.
I'm not sure if that's a good answer, but
Is the possible answer, so it's good. Another one, in your experience, is it possible to analyze oxidative damage removal using alkaline Comet assay without using FPG enzyme? And if so, yes.
well, oxidation damage does include strand breaks. So looking at recovery from hydrogen peroxide damage could be seen as measuring oxidation damage removal without FPG, where you'd be looking at the reduction in strand breaks after hydrogen peroxide. But if you want to detect the oxidized bases as well, you do need FPG. or OG1, of course, the human or mammalian equivalent. And in terms, the rest of the question was to do with
The recovery times.
And I would say strand break repair is rapid. As you saw, it's completed within an hour, so 10-minute intervals would be recommended.
I have more two questions. One is, how important is the moist chamber during the incubation of Comet slides with DNA repair enzymes?
I must say I've never checked this. I assumed at the beginning that it would be useful because it would prevent the gel drying out and the enzyme stopping working. If you use 12 gels and you're using the 12 gel chamber unit, the wells are sealed, so you don't need a moist chamber. The wells are effectively the moist chambers.
Okay. And the final one, at least from this list here, what do you consider the most challenging aspect of applying this assay for the first time? If you have important tips for
Well, the first important tip is to follow a standard protocol. And of course, I would recommend the various Nature protocols papers that have come out, which have very detailed protocols. Secondly, include controls, negative, positive, and others as specified in the protocols, and build up a historical, a database of historical controls. Specifically, if you're doing the in vitro repair assay, it's useful to make a batch of treated cells. In other words, freeze them as aliquot so that they can be used in many experiments, and you have the assurance of identical substrates for all samples. And this is especially important in biomonitoring. But Maria, do you have anything to add to that?
I think you you covered it. I will talk about historical control, needs of historical control and reference standard. How important is to keep the quality and kinds of competence of build the competence of laboratory. It's important, and from start, the people don't check what kinds of level of damage, or in this case repair was background level of repair was in a year before or 2 years before they started, and that might be showing if they have good quality from start, or they can improve, and then the standard deviation or variation coefficient goes more acceptable. Within 10%, it would be good.
Okay, are there any other questions from the audience?
From the chat, it was just that one. And
Yeah, no, we have a question from Marco. No, because he's just talking about, it's not a question.
Okay, so I'll hand over to Maria.
Yes, I will like to say thank you again, Andrew, for your presentation and discussion, fruitful discussion. And now we will go to our second expert, Dr. Maria Dusinska. If you want, you can share already also your screen, and I will introduce you, but you will not need introduction, but here I go. So Dr. Maria has a long experience in environment and health field, hazard and risk assessment, genotoxicity and carcinogenicity, nanotoxicology, advanced in vitro models, human biomonitoring, and biomarkers. Before she moved to Norway in 2006, she worked at Slovak Medical University as head of the Department of Experimental and Applied Genetics in Bratislava. At NILU in Norway, she was director of the GLP laboratory and scientific leader of Health Effects Laboratory. She was also teaching at Oslo University food toxicology and in Comenius University DNA stability and human health. In 2005-2006, she was national expert working at the European Commission Directorate General Research and Innovation for Health at Brussels for alternative toxicity tests. Since 2013, she's a member of the Scientific Committee on Consumer Safety, and since 2020, member of Genotoxicity Cross-Cutting Working Group in EFSA. She's editor of Mutation Research, Nanomaterials, Frontiers in Toxicology, and guest for many special issues. She coordinates several European Commission projects, and she continued to be a partner and working package leader in many other European Commission projects, namely the PARC project, that have a great interest in that one. So thank you so much, Dr. Maria, for accepting this invitation and share your knowledge with us.
Thank you, Karina, for very nice and quite long introduction. It's my pleasure to be here and to tandem Andrew with the presentation. I will be more practical, perhaps, and more to the ground, and try to explain how the Comet assay can be used in hazard assessment of chemicals and nanomaterials, and in genetic toxicology generally. So the Comet assay is really very known and much used assay, and with many models from in vitro, in vivo, and plants and humans. And I would recommend this article already mentioned by Andrew in Nature Protocols, which show several modifications of the Comet assay and includes many protocols. and it's actually the one of the most used assay for testing genotoxicity of chemicals, and I can say the most used assay for testing genotoxicity of nanomaterials, and specifically in standard in vitro cell cultures. But more recently, there are many applications for co-cultures, especially liver, lung, and skin. But even this assay is used so much, how the regulators accept it? In the risk assessment, it's important. Genotoxicity is one of the most critical endpoints in hazard identification, because if it can give you yes or no answer, and it's basically this genotoxicity endpoints is used in across all legislations, depending on the application field, and also depending on routes of exposure. And we have inhalation, ingestions, dermal exposure, or injection. But is actually Comet assay accepted by regulators? When we look at the EFSA Food Safety Authority, so which genotoxicity battery is used, it's for in vitro assay, gene mutation, Ames test, and chromosomal aberrations. I must say that it is of course basic knowledge that these are endpoints which are definite and not reversible. So these are the most crucial endpoints genotoxicity endpoints to investigate. And in case of nanomaterials, it's not Ames test, but mammalian gene mutation test. I don't want to go to the details. So if one of these tests is positive, then it's a follow-up, which includes in vivo test. And you could see here that among them is in vivo mammalian alkaline Comet assay, which has OECD test guidelines for 489, and it was adopted already 7 years ago. And even more, it's now usually chosen as endpoints for gene mutations. And the reason for that is practical, because it can be investigated in any target organ, and also in combination with micronucleus assay. But also David Kirkland and many other scientists, they did review and reanalysis of data from transgenic rodent somatic and germ cell mutations compared with in vivo Comet assay, and found very good correlation. So in vivo Comet assay is accepted by regulators in hazard and risk assessment. When it comes to consumer safety committee for mostly cosmetic, but also some other consumer products, again, it's a two-test strategy, gene mutation and combination with chromosomal damage, and usually bacterial gene mutation test and micronucleus assay. And again, in case of nanomaterial, bacterial gene mutation test cannot be used, so it's mammalian gene mutation test. And in follow-up, you can't use in vivo experiments to test cosmetic products, so in cosmetic regulations from 2013. So here in vitro Comet assay can be used in standard 2D model or 3D model, but only as a weight of evidence. And the reason is that there is no OECD test guideline for in vitro Comet assay. There is a test in test program project 3D skin model, micronucleus and Comet assay. I will mention it later, but also other new advanced model methodologies such as gene expression, reporter genes in silico can be used in weight of evidence to exclude genotoxicity, or to find a reason to exclude genotoxicity for specific chemicals. Recently, I would like to mention that EFSA issued last month this report, which is a harmonized approach for reporting reliability and relevance of genotoxicity studies. And this is showing how actually scientific committees evaluate publicly available the results from the all genotoxicity tests, including Comet assay. So as I said, there are so many publications on different chemicals and nanomaterials tested in Comet assay. Not many are actually taken into consideration when it comes to weight of evidence, because weight of evidence approach is also used in EFSA. So this is based on Klimisch criteria on reliability, relevance, and relevance. And there are scores. Score 1, reliable without restriction, only applies to test guideline and GLP studies, or in some exception also scientifically validated protocols, but this is, for example, case with nanomaterials. And it's a practical reason for that, because there are not many available studies in with other assays. But then reliable with restriction applies to scientifically validated methods, which are well performed. Only these two, reliability 1 and 2, can be taken further for investigation of relevance. And there is a relevance of test system and relevance of test results. So for relevance of test system, it's you could see that only Comet assay in vivo is considered, and Comet assay in vitro with other genotoxicity tests is giving lesser relevance or limit, what we call limited relevance. There are some exceptions with Comet assay in vitro as well, but it is case by case if it is route of exposure and well performed study, etc. So relevance of the study results depends on route of administration. So for example, oral is relevant, or CACO2 cells are relevant for foods, compounds or ingredients included in food. Biological relevance of the study results, it depends also from the purity of the test substance and specification of the substance, the other characterization, metabolic capacities of the system, etc. So when publishing the data on Comet assay, one must be very careful also to include all essential information about the tested compound, otherwise it will be excluded or giving low relevance. And I want to stress that studies which have a low relevance or are evaluated as having a low relevance are not considered in weight of evidence. And there is also quality, which is important, reproducibility of study results, reliability of study general, and especially cytotoxicity. If the compounds or concentration tested are cytotoxic, of course, it cannot be considered as a valid when high level of DNA damage is detected, because it could be interfering with high cytotoxicity. So how to ensure that Comet assay study is considered reliable and relevant? And it is then essential to properly design, perform, record, and report the study. So it could be done properly, but if you don't include the essential information in the publication, it could even paper from very highly reliable lab or with good results can't be considered relevant because, for example, there is no purity of the chemicals and other reasons, such as, for example, if there is no OECD test guideline or scientifically validated protocol mentioned, probably model is not relevant from the route of exposure point, or it's not sensitive. There is no reproducibility, and also standardization level of the test model. Then, of course, the timing, treatment, usually short and long, is recommended, and concentration range, which should be in a non-cytotoxic range. So cytotoxicity always needs to be included in cytotoxicity data in publications. There is a case in many cases this is not actually sought. And as I said, specification about the chemical or nanomaterial tested, characterization of the pristine material or chemicals, and also information about solubility and treatment behavior in treatment medium. And in case of nanomaterials, it should be also accompanied with cellular uptake, or at least it should be a reference to the data showing that nanomaterials or chemical is bioavailable. So is it reaching the cells? And as Andrew already mentioned, absolutely crucial, positive, negative, and also in case of nanomaterial interference controls and reference standards, and especially historical positive and negative controls. These are hardly you can find in any publication. And the laboratories are doing Comet assays around more than 3 decades, almost 4 decades. So there are data available in laboratories, but they are not reported. And then, for example, if the level of background damage is so low, and you don't provide historical data that these cell lines have this range of background level, it is the reliability is questioned, and of course it influences relevance. So additionally, essential technical information on Comet assay procedure needs to be mentioned, such as pH of all solutions, concentration of agarose, lysis time, incubation with lesion-specific enzyme, and if this is used as a modified Comet assay for detection, for example, oxidized DNA lesions, and alkaline unbinding time, electrophoresis conditions, and time of electrophoresis, and also voltage gradient, volt per centimeters. And stain scoring method, Comet descriptors. It's also usually people love olive tail moment or tail moment, but regulators or scientific committees, they don't really see what's the level of damage. It's just number. So percentage DNA in tail intensity is preferred. And of course, statistical analysis, it's must. So you should have enough replicates and number of experiments, and then not only statistical significance in p-value, but also trend. So if there is any concentration response, if this is for in genotoxicity studies, and it should be clear criteria for positive, negative, and equivocal. This some researcher, they just judge in their own way that this is positive and this is negative. But usually, as in other assays, at least double damage 2 times higher than background level. It's absolutely a must, and then it should be at least one significantly higher concentration plus dose response or concentration response. In actually nanomaterials, it's not always concentration response achieved. So in this case, at least 2 positive concentrations could be attributed to positive response. And also, it is a study done in GLP compliance. It's usually considered by risk assessors. And these are 2 papers which are published in scientific literature. One in Nature in Nature Protocols, and the shorter one as editorial from Gunnar and Andrew, which actually summarizes most of the point I already highlighted. So I highly recommend all researchers, not only those who are doing genetic toxicology studies, but in all Comet applications to read and apply them. So how to report raw Comet data? I think reporting raw Comet data is depend on labs. Everyone has own style, but what is actually needed is a harmonized way for data reporting and data presenting. So we in RISGON we developed in collaboration also with other projects template wizard, so harmonized template which for reporting data not only for the Comet assay, for over 60 other methods in not only in ecotoxicology, genetic human toxicology, but genetic toxicology and other assays, but also in characterizations and analytical methods. And this is actually accepted for publication, and it soon will be published. This is also in Nature Protocols paper template. These are co-creation of data collection templates for safety assessment of nanomaterials, but actually it was already applied in many other different fields. It was created in collaborations with experimental experts, with IT experts, and the reason was also to have a good to create a good database which with nanomaterials in eNanoMapper, and also to allow following fair data treatment to reuse the data and to do meta-analysis, and these regulators and risk assessors would highly appreciate. So when it comes to Comet template for reporting Comet assay, you could freely download it from website. This is freely available, and you go to the data template entry, and then click the Comet assay, download the template. Here is the link. And this is online available, but you can also use it offline for reporting data. It doesn't need to be together with InanoMapper. It is excellent tools to harmonize the way for data reporting, and it is based on Excel. So first sheet actually force you to give all crucial information which you will later need in preparing of publication. And the second sheet, it's reporting for raw data, which automatically calculates median and then means of medians, and also generate the figures. Of course, these templates could be slightly modified, or can be modified by those who are using them for different reasons. But as a template, as a first template, it's really good to use it. So if you don't have a chance to download it, it's available at Nilu Laboratory, and we can send it to you. So first, we actually see that Comet assay is not fully acceptable by in vitro. I'm talking about Comet assay in vitro now. It's not fully acceptable by risk assessors, only in weight of evidence. But it's getting more and more important to know for next generation risk assessment, which will be certainly without testing on animals. It is important to know mode of action, and in this case, Comet assay can be very important assay, especially to detect specific DNA lesions such as oxidized bases or alkylation, and even more important for not only for primary but also for secondary genotoxicity. And I will highlight why the 3D models can be useful for this. So just to remind to all of us that mode of action for genotoxicity, there are several ways how DNA can be damaged and can lead to gene mutations or chromosomal damage, chromosomal aberration, or anogenicity. So it could be direct contact with DNA, or indirectly, which is in most cases, through oxidative stress, cell membrane damage, mitochondrial dysfunction, antioxidant depletion. So there are many events which could lead to indirectly to DNA damage, and this could be captured with especially with the modified Comet assay. And also DNA repair is one of the reason if it's not in normal function. When it comes to secondary genotoxicity, it is done through other cells, through oxidative DNA ADAC by reactive oxygen species, through activated phagocytes during, for example, nanomaterial induced DNA inflammations. So here you could see directly, or at least there is a debate if nanomaterials enter the nucleus, but definitely they enter the cells. But here it's nucleus, and we can see nanomaterials in. They could also enter through cell division. So knowing that, it is important, and when we say that there is a secondary genotoxicity, which is in vivo in most cases, so it is mediated via the other cells, mostly phagocytes, but also the others. Then 3D models are crucially important to be established in next generation of risk assessment. And one of them is actually already validated, and it is 3D skin model. And it's a combined model with in vitro alkaline Comet assay, as well as micronucleus assay. And this is actually already ongoing from 2019 OECD project. and it's validated. It's just need adoption with and agreement of all OECD countries. And these are two papers. There were so many which already show that this combined model, alkaline Comet assay and micronucleus in reconstructed 3D full skin model is validated. And other very promising new advanced models are 3D liver model, which can actually say about can give information on mode of action, especially when it is the systemic exposure. So in our our laboratory, our PhD student Elizabeth, who's actually a few weeks ago, she successfully finished her PhD. She established this hanging drop model of liver spheroids, and also adapted it for the Comet assay and micronucleus assay. So here are a few publications from her, which were basis for her PhD. And also she focused on advanced bronchial and alveolar mono and co-culture models. Also here in this graphical presentation, there are 4 different mono and co-culture models, BS2B cells, alone or in combination with endothelial cells, or A549 cells alone or in combination with co-cultured with endothelial cells. And you could see that it is on a transval membrane. The lung cells are on the top and the endothelial cell on the bottom of the membrane. But actually, when they are exposed with in aerosol, in cloud with nanomaterials, these are results in with hydrogen peroxide, which show actually that BS2B cells are the most sensitive cells, and both in monoculture and co-culture with endothelial cells. And these are results on nanomaterials, and these are BS2B cells. But we also have results on endothelial cells, which are on the other side of transfer membrane, where actually we can see the effect. So nanomaterials sits on the top layer, but still effect can be seen also in lower. So these are one paper in collaboration with another group in Oslo, and I unfortunately don't have a picture of Laura, and again Elizabeth papers. So how we can contribute to standardization and validation of Comet assay, and that means both for standard in vitro model as well as co-culture models. Within RISGON, we actually focus on standardization and validation by evaluating, optimizing, and pre-validating several SOPs and also test guidelines, specifically focused on nanomaterials, because test guidelines are adapted or developed for standard chemicals. And we use the harmonized approach using the same nanomaterial characterization, dispersion, and interlaboratory comparison with many assays. But here I only highlight for environmental toxicity, genotoxicity in Daphnia, Comet assay, and also what we performed in different cell lines for the Comet assay, but also other assays, genotoxicity test, micronucleus, and mammalian gene mutation test. So the huge effort was done in our laboratory. So these are members of health effects laboratory at NILU. But also I can't put pictures of all who were included from RISGON. There were 4 laboratories doing or involved in Comet Assay, and starting with discussing and methodological adaptation if needed for standard operating procedure, then optimizing it and testing with in series of interlaboratory studies. And then if another adaptation was needed, so it went through another testing interlaboratory study with selected panel of different nanomaterials. And the aim was to select the most promising methods, which one of them was Comet Assay, and translate it into so-called SPSFs, which is the application for the project in OECD test guideline program. So these are some of the results. I am not sure if all are evaluated, and there was acceptable correlation in some cases, but in some cases still some improvements are needed. And the Comet Assay, the miniaturized version of enzyme modified Comet Assay for genotoxicity testing of nanomaterials, which we applied in this project, was published. But as I said, you never do any Comet Assay without cytotoxicity study. So in coupled with this, we published 2 cytotoxicity assays, Alamar Blue and colony forming efficiency assay, which is actually very promising for testing nanomaterials, which at least the colony forming efficiency is also promising to be developed as an OECD test guideline. So which methods are currently in OECD test program? It's a tox tracker, 3D skin model with reconstructed skin, micronucleus and Comet Assay. These are actually, these 2 passed the validation, and now there is a peer reviewing ongoing, so hopefully soon they will be adopted. And there is also in vivo Comet Assay initiated from Norway and Nordic countries, Comet Assay for gonadal cells, a specific genotoxic effect. It just started last year, and there are several other assays, but you could see there is no in vitro Comet Assay with standard cultures or other co-cultures yet. So I just leave this question open. What is needed for Comet Assay to be formally validated and accepted as OECD test guideline? I have some answer, but they could be more efforts needed, resources needed, communications with national coordinators, which are representing each country, and lots of enthusiasm from Comet community. Thank you very much.
Thank you very much, Dr. Maria. I would like to ask now to the audience if somebody have any question that want to do directly to our expert. In the chat until now is nothing. So probably I will start with some questions also that were addressed in the Google Forms during the registration. Okay, there is here one question in the chat from Roger. Do you think regulators should give a different weight on data obtained with the normal Comet Assay compared to data from the enzyme-modified Comet Assay, because the latter will give information on the type of lesion?
Thank you. That's very good question. And as I am working in two scientific committees for risk assessment of cosmetics as well as EFSA, and of course the scientists, and this is I would like to encourage experts to join these committees, because this is the way how you can translate knowledge directly to regulators. In these both committees, we always, if data are available on the modified Comet assay with lesion-specific enzymes, especially with FPG or OGG1, we take them into consideration in weight of evidence. But many of these data are not giving. Some of these are giving lesser relevance because of data reporting issues. And for example, I know many laboratories, I know they are good, but it's very difficult to give the good relevance or high or limited relevance if this essential information is not there. But definitely, as a weight of evidence, also DNA oxidation is considered, and also it's much more valued if the results are both for strand break as well as oxidized DNA lesions.
Okay. So I will now make a question that were previously made in the Google Forms. Since most agencies are moving away from in vitro assays and often encouraging in vitro testing and NAMs, how do you see evidences from in vitro Comet assay results being integrated?
I think I answered already these questions. It's important to further develop the Comet assay in 3D models because of relevance for route of exposure and secondary genotoxicity. And also, I think it's important for standard in vitro Comet assay because with the modifications, including DNA repair, can be used in mode of action and as a weight of evidence, and also in next generation of risk assessment. Every in vitro test, and both standard and co-culture and advanced, is NAM, including in silico. So everything what is done in vitro or ex vivo, not on animals, is NAM, including human biomonitoring data. So even human biomonitoring data will become more important when assessing safety of chemicals and nanomaterials. But that's future, because we need to, and it should be not only standalone test, but the combination of information from even different models, different assays to see whole picture and possible mode of action. And especially with advanced outcome pathways and to identify key and events and
yes, the AOPs that are also NAMs. Another question: can an approach such as creating genotoxic units with DNA tail density or moment data be used created in risk assessment?
Well, actually now in our committees, when it's olive tail moment, and when you don't see what is the baseline level of DNA damage, because that parameter just is number and doesn't tell you what's the level of DNA damage, but percentage DNA in tail gives you this. So it is accepted by risk assessor as unit. But of course, it could. There was in past Andrew and also we actually together were able to, and Gunnar Brumborg, translate the percentage DNA in tail on number of breaks per Daltons and per cells. So it is possible to quantify the damage.
Another question: how sensitive is the Comet assay for detecting DNA damage in lymphocytes from smokers?
There are lots of human biomonitoring data, including those which we were doing when I was working in Slovakia. In some studies, we found that smokers have a higher level of DNA damage, but mostly it was in correlation with other markers, for example, level of vitamin C. Those which have a lower level of vitamin C were smokers and that they had a higher level of damage. So in this case, some studies they don't show it, and I think reason is that Comet assay is sensitive and of course can capture or can be results of overall exposure. So it's not just smoking. So and it's a cumulative exposure and it could be aggregated exposure. So and smoking is additional to all this. So but it depends also design of study, how you select your group.
Yes. So there was another question that you already answered. It was about the human sampling after exposure, because it was more or less common for both of you. So I will skip to the next one. What should be taken into consideration when performing Comet analysis from buccal cells?
I think it would be extremely good to have this method standardized. And I again want to refer to the Nature protocols, this compendium of protocols. It of course will need additional step to digest the cell membrane, because of course cell membrane is much tougher. It needs to be, because buccal cells are directly in these are directly actually
Exposure contact.
Connected with exposure. But for example, for children monitoring, it would be extremely good to have this non-invasive way of sampling, and also to sample target cells. These are target cells, for example, for food. And the same is for eye and nasal buccal cells as well. So it's this is the critical step to digest the cell membrane, but there are details in Nature protocols about this.
Yes. There is a question asking, maybe is one of your co-workers, I don't know, let me see. Are there plans to write a manuscript that involves standardizing Comet assay using cell culture?
Yeah, I will tell them yes, it's in my list.
I don't know who wrote, but I was thinking probably one of co-workers that is the job to do. So are you planning?
Yeah, they don't have time to do it. So there is actually there are very good groups in Europe. For example, Swansea, who are very good in liver model with micronucleus, and I think the NILU group is very strong with Comet assay, but also started with micronucleus, and also there is a group in Ireland. And we already started discussion to do interlaboratory studies to have a liver model for both Comet and micronucleus, and to be prepared in future for OECD test guidelines. And of course, you need to first prove it scientifically. It should have a scientific acceptance, and then it can go to. It should be standardized and pre-validated by the scientific community or researcher, published, and then based on the quality of papers, then could be considered for further development of OECD test guidelines. So yes.
I have a more general one, because this is in literature. We will see that there is already a lot of Comet assay studies in the exposure to pesticides. So this question is, can the assay help to assess toxic effects of pesticides in farmers?
Yes, definitely. And it was done. There are many papers on this.
Yes, there are also systematic reviews and meta-analysis regarding this in particular.
Yes.
more two questions. Do you think that buccal cells can be used for biomonitoring a population of radiation workers with alkaline comet assay, such as hemodynamists who are currently exposed to low doses of ionizing radiation, sorry?
Yes, but also systemic exposure actually can be captured by human blood. So.
Mm-hmm. So you would say if you have to choose one biological matrix, would be blood instead of buccal cells in this particular...
But it could be nasal buccal cells. Yes,
Okay.
definitely yes.
The final one, at least from here. Comet is considered to be a versatile but complex tool in genotoxicity testing. Why is that? Can we overcome it?
So I am not sure if I completely understand this question. It is the assay which can measure several endpoints, which is very, very important. And it's very versatile because any cells can be used and it's really used in different fields. So if complexity is taken from this point of view, yes, but how to overcome? No, I wouldn't like to overcome it. I see it as an advantage, of course.
I don't know, because I don't know the rationale behind the question, but to me, I think that, okay, it's a versatile technique. I think the complexibility as a tool in genotoxicity testing, I suppose that is Comet assay is classified also as a biomarker of exposure.
Yeah, definitely it's not biomarker. It's mostly biomarker of exposure. When it comes to, it could be considered, it's on the borderline, but definitely it's a marker of exposure. And because it's a lesions are transient and can be repaired.
An effect.
So
like micronucleus.
Yes. So micronucleus and gene mutation. That's the definite. DNA, fixed DNA damage into this. So from this point of view, yes. But on the other hand, and this is why regulators do not take it first, because it's no OCD test guideline. It's only in weight of evidence mode of action. But for example, in vivo, Comet assay is really in a highest level as the micronucleus in bone marrow or gene mutations in vivo, mostly from the practical reason, and even it could be transient DNA. The lesions are transient, but it's a high correlation or high probability that they could lead to gene mutation.
Yes, I suppose it is because of that. Because
yes. So how to overcome this? The modified version of the Comet assay, I think it's more specific. Let's put it like that.
Okay. There is questions in the chat. So again, Ali, what are considerations to be taken when checking for DNA damage or repair in heterogeneous mixture of cells like whole blood? Is one question. And can this impact the validity of my results, knowing that certain cells could be more susceptible to damage than others within the same sample in the case of whole blood?
Yes, there are many cells, and not only lymphocytes, but others, but depends what is the hypothesis or what is the question. If it is question that this chemical or exposure can lead to DNA damage, I mean that's okay. You have a mixture of cells, but the majority of these are lymphocytes. In whole blood, as Andrew already said, it of course it could interfere with the proteins present in blood. And actually, with hydrogen peroxide, not in whole blood, but in lymphocytes, we found that there are two subpopulations of cells. One are very sensitive to the DNA oxidation and or hydrogen peroxide treatment. Another, they are resistant. So if you see the Comet images, you could see this very, very small damage and high damage cells. I think that is natural. So it would be good to monitor, actually, to present in publication as additional information that there are more sensitive cells in the blood. And we actually, in past, we were collaboration with immunologists, wanted to study subpopulation of blood cells to see which are most sensitive, or if there is DNA damage difference in background level as well as induced level.
I can add that, Ali, I don't know if you saw or first meet the experts, but because it was about different subpopulations in blood. So if you check there, is in YouTube, in our YouTube channel, you can see the differences in at least the DNA damage, not the repair, because it was strictly about the DNA damage. The differences of the whole blood and the isolated lymphocytes, advantage and disadvantages. Whole blood have all the type of cells. In the case, in leukocytes are cells with a small lifespan. So lymphocytes have a longer lifespan.
20 days, I think.
More information about cumulative exposure, but is 1 hour is something of session 2. So maybe your doubts can be there to see the things. So Juliana, it was the person who did the question about
and I see actually it's very interesting, and it would be good to collaborate, because in Health Effects Laboratory the focus is also on breast model, so it would be good to have collaboration in this matter. So please contact people.
Yes, is my final message. First, I would like to thank you, Dr. Maria, to join us in this.
It was a pleasure.
Thank you so much. And I want to show again my appreciation to our experts for accepting this invitation and be here with us. Thank you so much, Andrew and Maria. Thank you for all the questions and participation. So in the chat also saying that were great presentations, and thank you and for sharing your knowledge. I hope it was fruitful for everybody. I see that is already here collaborations that are starting with the 3D models. I would like to say that any question or doubt that comes to you, you can also email the experts. So their emails are very well known. In a Google search, you will find it for sure. And we had some questions that I not put here because regarding the falling out of gels, for instance, maybe you can see first the videos that we have in YouTube channel about the technical procedures and these papers that are already published with the good practice for the Comet assay. And if that persists, you can all the time contact us also. So again, I would like to remember that this meeting can be, it was recorded and can be revisited in the YouTube, in the ICAWG website. And I wish you all good experiments and good comments. And more than that, I wish you now a nice weekend. So thank you so much.
Thank you, and nice weekend to you too.
Thank you.
Bye.
Bye.
Bye, everyone.
Mm.